మనకు ఫ్లవర్స్ లోపల ఇన్సైడ్ ద ఫ్లవర్స్ stamens are present. Inside the flowers, stamens. Stamens represent the male reproductive part. Stamens is present with the stalk. A long part stalk is present, and in terminal part, that is anther, is present. Okay, the anther is generally two types. Bilobed anthers are present, monolobed anthers are the present. So bilobed anthers mostly present in the dicots. The plants normally what we are observed outside, they present with the bilobes. And that bilobe means two lobes are the present. Inside there is a two lobes are the present. Again, each lobe is present with the two thecas. Each lobe is present with the how many thecas? Two thecas. Okay na? There vertical and horizontal two thecas are the present. That is called dithecous condition. Dithecous condition. Okay na? Right? When you observe bilobed dithecous anther, it will present with the four chambers. Four chambers. There are four chambers are the present. 1, 2, 3, 4. There every chamber is called microsporangium. Every chamber is called microsporangium, or also called spore sac or pollen sac. Okay, right? So when you cut this anther, cut this anther into the transverse section. So the meaning of the transverse is nothing but the anthers are taken, anther cutted into the which one? Horizontally, transverse section. And after the cutting, if you observe under the microscope, there is some layers or surrounds to the that microsporangium. So this one is microsporangium, 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 microsporangium. So that microsporangium is present with the some layers. That layers makes the wall. Okay, the microsporangium wall is present with the what is four major layers are the present. There are four layers are the present. In that first layer, epidermis is the first layer, and endothecium is the second layer. Next, middle layers are the third, and tapetum is the fourth. So these are the four layers we can observe under the microscope when the microsporangium is cutted. Again, once again, the first layer is called epidermis, the second layer is called endothecium, third layer is called middle layers, and the fourth layer is called tapetum. Okay, now coming to the first part, epidermis. When you observe the epidermis, epidermis is the outermost layer which is present outside, outermost layer. And this epidermis is a single layer in the nature. How many layers? Single. Outer and single layer. And the cells are arranged in a closer to each other. Side by side arrangement is present and there is no gaps. But the epidermis at the corners of the microsporangium, where it is? Corners. Corners are present with some specialized, large sized, fan shaped, specialized, large and fan shaped cells are present, which known stomium cells. That stomium cells helps in the splitting of the pollen sac or splitting of the microsporangium. That dissensi we can call. Okay, right? Overall, the epidermis is helps to protect the remaining inner parts, and it is also helps in the splitting during the dry. Okay, this is about the first part, epidermis. Now come to the second part, endothecium. The second part of the endothecium is endothecium present below the epidermis. Endothecium present below the epidermis. It is mostly 2 to 3 layers. 2 to 3 layers. In this, cells are radially arranged. How it is arranged? Radially arranged. and cells are present with the fibrous thickenings. The thick walls are the present, fibrous thickening. And these endothecium cells can lose the water at the time of the maturity. At the time of this microsporangium maturity, it can lose what? The water. By loss of the water, it can be break. Okay, that nature is called hygroscopic nature. So the endothecium is present below the epidermis. It is 2 to 3 layered, and it is present with the radial cells. It is present with the fibrous thickenings, and this is can lose the water, which known the hygroscopic, and it is helps same in the splitting of the pollen sac or microsporangium. Then the third part third part middle layers. Just below the endothecium, there are 5 to 6 layers are the present. They are arranged in the middle, that's why we can call simply middle layers. Okay, now below the endothecium there are few layers are present which call the middle layers. The middle layers are present with the thin wall, simple thin wall is present, and they can also involve in the splitting of the microsporangium, because they will make dry and they will be helps in the splitting of the microsporangium. After that, there is a tapetum is present. The tapetum is the last part actually. Okay, now what are the points you can say to the tapetum? It is innermost layer. Tapetum is what? Innermost layer. Okay, now right. This is present with the thin wall cells. It is present with the thin wall cells, and it is having with the more cytoplasm. In this, cytoplasm is more. The nucleus, one or many nucleuses are present. One or many nucleuses are present in that. And the main nature of this is that is nutrients having tissue. It is having with what? Nutrients. By having of the nutrients, it is called nourishment tissue. Which tissue? Nourishment tissue. It nourishes, it gives the nutrient for the developing sporogenous tissue further. Okay, now the tapetum is supplies the nutrients to which layer? To sporogenous tissue. Okay, now so this is overall microsporangium wall layers, and the microsporangium wall layers are the these are the 4 types. Okay, now this is a topic very important for your 8 marks question. Okay, now the diagram should be drawn. After that, label should be done. After that, you need to explain the each and every point. Stop this.